Okay, this is series of GCSE question on transport in cells. So we're going to go through the questions quickly. Now, the first question here is, they said this question is about cells and transport. Now, they say complete table one, which is name of cell part and the function. So contain genetic information, that's the nucleus. Mitochondria is used for respiration to provide energy. They control the movement of substance in and out of a cell. That is done by the cell membrane. So I'll just be writing the initials. Cell membrane. This is quite straightforward. Now, cells in potato are plant cells. Now, cells in potato do not contain chloroplast, so they don't have chloroplast. Now, what is the function of chloroplast? Chloroplast help to they provide or produce or they make chlorophyll, and this chlorophyll is used for what? It's used for photosynthesis, to trap sunlight for photosynthesis. So that's it. Now, the next question here is, they said, name one type of cell in a potato plant that does not contain chloroplast. That's the root hair cell. The root hair cells do not have chloroplast. Now, a student investigated the effect of salt concentration on a piece of potato. This is the method used. Now, they cut 3 pieces of potato to the same size. That makes it, that's a control variable to make it a fair test. They record the mass of each potato piece. They add 150 cm cube of 0.4 mole per dm cube salt solution to a beaker. Now they place each potato piece into the beaker, and after 30 minutes, they remove each potato piece, dry the surface with a towel, and they now record the mass of each potato piece. Now they repeat step 1 to 5 for different concentrations. So what was changed in this experiment? So what is changed in an experiment is your independent variable, and what is being measured is our dependent variable. So what was measured was the initial, so they record the mass of each potato at the start, and they also record the mass after 30 minutes, which is at the end. So that is your dependent variable. But your independent variable is the change, the different concentration of salt solution, because that was changed in the experiment. So the first question is, what is the independent variable? So that would be concentration of salt solution. Now, why did the student dry the surface of each potato plant with a piece of towel? Simple. Now, that is, they do that to make sure only the potato mass was measured. That means only the potato mass was measured, because if the water is left behind, it will give it a higher mass. So the mass of the water will be added to the result. Now, the student calculated the percentage change in mass of each potato. Now, for one potato piece, the starting mass is this and the end mass is this. So if we want to calculate percentage change, they give us the formula here to use: increase in mass over starting mass. So your increase in mass is the final, I used to call it final minus initial, or the new minus old. So the final value here, which is the end mass, is 2.7. So it will be 2.7 minus 2.5. 2.5 divided by starting, which is 2.5, divided by 2.5 times 100. So once you do that, you get your answer. So that's it. The answer should give you 8%. Now, they said the student used the result from each potato piece to calculate the mean percentage change in mass at different concentrations. So you have this table here. They now say we should complete figure 1, which is this graph. You should label your x-axis, use a suitable scale on the x-axis, plot the data from table 2, and draw a line of best fit. Now, first, if you need to understand that when reading your letters, your x always comes before your y. So the skill there is that what is written first, which is your independent variable, and what is recorded, your dependent variable. So always your independent variable from your x-axis. So the first criteria here is for us to label it. So when you want to label it, everything that is written on this heading here, you should just copy it down. So what I'll do on my x-axis, I will come here and I'll write concentration of salt solution in mole per dm cube. Now, once that is done, the next criteria, so I'm done with this first criteria. The next criteria is to use a suitable scale. So look at it. It makes it easy for you. Now, it is 0, 1, 0 0.1, 0 0.3. So I will make sure your scale covers more than half of the space given. So if I make this 0 0.0 and this 0 0.1, and I have 0 0.2, I have 0 0.3, I have 0 0.4. So it covers most of it. So the next thing is to just plot your data. So at 0 0 we have 9.8. So at 0 0 I have 9 here. So this is 5. So this will be 9.2, 9.4, 9.6, 9.8. So I will put a star. At 1, I have 9.5. So at 1, here we have 9, 9.2, 9.4, 9.5 will be somewhere here. Then at 0 0.2, I have 7. At 0 0.2 is 7, which is here. Now at 0 0.3, it is 0 0.4. At 0 0.3, I have 0 0.4. So this is 0, 0 0.2, 0 0.4. Then at 0 0.4, I have minus 1.4. So if this is minus 1, minus 1.2, minus 1.4 will be here. Now, I'm told to draw a line of best fit. So I'm done with plotting also. I'm done with the scale. I'm done with plotting. Now, the last thing is line of best fit. So the line of best fit should be drawn to touch as most of the what is it called, plotted point in a straight format. So what I can do, let me see if these three will join. I think this should be able to go down. Sorry, you have to make sure you use your ruler to make sure it's straight. Now that's it. Okay, however, they said what concentration of salt solution was equal to the concentration of solution inside the potato cell. So at that point where it is 0, where it passes the line, your line of best fit passes 0.0 here, so that will tell you the concentration. So if it passes it here, we are having around, you make sure you mark it where it touches it. Then if you draw it down, this is around 0.3. So this is 1, 2, 3. So 0.3, sorry, what am I saying? So 2, 4, 6, 0.36. That would be the answer. So now they said explain why the potato piece in the 0.4 mole per dm cube salt solution decrease in mass. So why will the potato decrease in mass? It will only decrease in mass if water moves out of it. So water moves out of the cell or the potato by osmosis because the solution in the cell have what? Is less concentrated than outside. So that means the cell have a higher water potential than outside. So water tend to leave the cell outward. So the cell will now decrease in mass. Now, here they said water and carbon dioxide are exchanged between the leaves and the atmosphere through the pore spaces called stomata. Now, name the cell that control the opening and closing of the stomata. That's your guard cells. I think this is, I put them there. Yeah. Now, water moves through plants in the transpiration stream. Yes. Now, describe two differences between transpiration stream and translocation. Now, transpiration is done by xylem, while translocation is done by phloem. Now, transpiration is have to do with water, helps in the movement of water, while translocation helps in the movement of sugar. So, you see, transpiration stream involves xylem, while translocation involves phloem. Transpiration transport water and mineral ions, while translocation transport dissolved sugar. Transpiration moves substance upward from the roots to the leaves, while translocation moves substance upwards and downwards. So they are bidirectional. Now, which environmental condition will cause the rate of transpiration to be greatest in a plant? Now, transpiration is the loss of water. Now, if you look at humidity, how do humidity affect transpiration? So the higher the humidity, the lower the transpiration. Because humidity is the amount of water in the atmosphere. So cold, what are we looking at? Which environmental condition will cause the rate of transpiration to be greatest? Now, so we need a lower humidity with warm temperature. So low humidity, warm temperature, that's the answer to have high amount of transpiration. Now, figure 7. Figure 7 shows the information about the mean width of the stomata in a plant. So we have midnight, midday, and midnight. So now the changes in the mean width of the stomata in normal conditions are an advantage to the plant. Explain how. So in normal conditions, the changes. So you see the normal condition here is this straight line, while low atmospheric carbon dioxide is the dotted line. So why this normal condition have an advantage is that at midday, the sun is at high intensity. So you find out that the stomata is open more at midday. Why? To absorb more sunlight that will be used for photosynthesis and the rest. So your answer will look like this. Stomata close at night because there is less light for photosynthesis, while stomata open more at midday for maximum light intensity so that carbon dioxide can diffuse in for photosynthesis. So your answer should be structured in this format. Here, where stomata close at midnight because there is no light for photosynthesis. Then closing stomata reduces and prevent water loss. But however, stomata open the widest at midday as maximum light intensity and carbon dioxide for photosynthesis. Now, the changes in the mean width of the stomata in low atmospheric carbon dioxide are different from the changes in normal condition. Now, explain how the difference help the plant to survive in low atmospheric carbon dioxide. Now, stomata are open wider and for more time. So it allows the plant to take in more carbon dioxide for photosynthesis. That's just it. So it will open for a longer time in areas with low atmospheric carbon dioxide. Now, oh, I didn't even. Okay, so diffusion is an important process in animal and plant. So define diffusion. So diffusion is the movement of molecules or particles from an area of higher concentration to an area of lower concentration. So we have figure 2 here shows part of a leaf. Okay, now we have our guard cells, we have our stomata, we have our mesophyll cells, and that is the part that is being displayed here. Now, so we are told that molecules of carbon dioxide diffuse from the air into the mesophyll cells, yes, which two changes will increase the rate at which carbon dioxide diffuse into the mesophyll cells? I think I've ticked the answers here. So increase carbon dioxide concentration in air. If there's higher carbon dioxide concentration in air, that means there will be low carbon dioxide concentration in the leaf. So carbon dioxide will now move by diffusion from the air, which is higher concentration, to the leaf, which have low concentration. Two, if there is increased number of stomata that are open. So the more stomata that are open, the more gas can move into the leaf and out of it. Now, I have this 6 mark question here, and the answer is also here, so we can just run through it. Now, diffusion also happen in the human lungs, and this is a diagram displaying the human lungs. So you know that this is the alveoli, and the alveoli have a lot of blood capillaries. It is one cell thick. It is also thin to make it efficient for diffusion. So they say, explain how the human lungs are adapted for efficient exchange of gases by diffusion. One is that it has many alveoli, which are this small. So these alveoli help to give it a large surface area to volume ratio, which make it efficient for diffusion. Now, a lot of capillaries around it. So capillaries are thin. So you have a lot of capillaries around the alveoli, and they help to keep or to maintain the concentration gradient for diffusion. And they also supply here with supply the alveoli with blood vessels to make exchange faster and provide a short distance for the exchange of oxygen and carbon dioxide. Now, it has a large capillary network, also good supply of blood. This helps to remove oxygenated blood quickly, to bring carbon dioxide to the lungs quickly, to maintain a concentration gradient. So the basic thing you should look at here is a 6 mark question. So alveoli, 1 mark, provide large surface area to volume ratio to make diffusion efficient, another mark. Now capillaries, it has thin walls to reduce the distance and make it more efficient for diffusion of substance. We give you another 2. Then breathing mechanism, so ventilation, it is supplied with a lot of air constantly, so you breathe in fresh oxygen and you breathe out carbon dioxide, so that will help to keep the concentration gradient efficient. So that alone will give you another 2 marks. So not that you have to write everything here, but each point you give and you explain, you should be able to hit it. Now, we are told that figure 4 shows a root hair cell. Now, name the process by which water molecules enter a root hair cell. So, water molecules enter a cell, root hair cell, by osmosis. Now, nitrate ions need a different method of transport into the root hair cell. Yes, they are transported by active transport. Explain how nitrate ions in figure 4 are transported into the root hair cell. Use information from figure 4 in your answer. So name of the process is active transport, and explanation, it is because energy is needed for this movement of ions. That's one. Also, because the soil have a lower concentration and the root hair cell have a higher concentration. So in this case, it is moving against the concentration gradient from a region of low concentration to a region of high concentration. So if you look at it, the root hair cells have more of this nitrate ion than the soil. So it will only be able to move by active transport. Now, here we are told that a student carried out an investigation using chicken eggs. Now, this is the method the student used. They placed 5 eggs in acid for 24 hours to dissolve the eggshell. Okay, now they measure and record the mass of each egg. Now they place each egg into a separate beaker containing 200 cm³ of distilled water. Now, after 20 minutes, remove the egg from the beaker and dry them gently with a paper towel. Now, you measure and record the mass of each egg. So this is the mass without shell, and this is the mass of the egg after 20 minutes in grams. Now, they said another student suggested that the result for egg 4 was abnormalus. So abnormalus, that means it does not follow the trend. Now, do you agree with the student? Give one reason for your answer. So, yes. Why? Because the mass change of egg 4 is much lower than the others. So, the change in mass for egg 4 is lower than the others. The others change in mass is quite higher than it. So now calculate the percentage change in mass for egg 3. So if you want to calculate the percentage change in mass for egg 3, it's quite simple. So it will be 75.7 minus 72.4 divided by 72.4 times 100. That's it. So which is the final minus what? The initial divide by your initial times 100. So that will give you 4.6%. Now explain why the masses of the egg increase. Mass of the egg can only increase as a result of osmosis. So that's it. Mass increase because water is entered by osmosis. Now, it's a 3-mark question. So if you just state water entered by osmosis, the maximum mark you get is 1. So once you write water entered by osmosis, you need to now explain osmosis itself. Now, so you see what the examiner wants to see. From a dilute solution in the beaker to a more concentrated solution in the egg through a partially permeable membrane. That's it. Because here we are told that the beaker has 200 grams, sorry, 200 cm cube of distilled water. So you find out that it is less concentrated, is dilute. So water tend to move from dilute solution to more concentrated solution. Now explain how the student could modify the investigation. This is the key word here. Modify the investigation to determine the concentration of the solution inside each egg. Now, so they can use more eggs, they can use more different concentration of salt, so you get more data. They can use like 5, 6, 7. Now, salt solution, or they use sugar solution, whichever, in beaker, and by plotting their percentage change in mass, you can use a graph to determine the concentration where the curve crosses the 0% line, like what we looked at earlier. Now, chicken eggshell contain calcium. Calcium ions are moved from the shell into the cytoplasm of the egg. Now, table 5 shows information about the concentration of calcium ion. Now, in eggshell and in the cytoplasm. So, eggshell 0.6 by cytoplasm 2.1. Explain how calcium ion are moved from the shell into the cytoplasm. That process too is by active transport because ions are now involved. So ions are moved from a region of low concentration to high concentration by active transport, which requires energy from respiration. That's it. So thank you. I hope this will help you in preparation for your mock exams.